Hello students, now we are going to discuss about biosynthesis of carbohydrates. Carbohydrates are found as monosaccharides, disaccharides, polysaccharides or complex carbohydrates. They are an important source of carbon for synthesis of other compounds and of chemical energy for immediate release as with glucose or fructose or for storage in the form of polysaccharides such as starch or glycogen. Many carbohydrates such as cellulose and chitin serve as structural function, providing support and protection. The term saccharide derives from the Greek word saccharon, which means sugar. Structural polysaccharides are frequently found in combination with proteins, that is glycoproteins, and proteoglycans, or lipids, that is glycolipids and lipopolysaccharides. They are also found in combination with peptides in peptidoglycans. Many of them, but not all, have general formula CnH2O into M. Some, in addition to carbon, oxygen and hydrogen, include nitrogen or sulfur. Later it was found that some of them, such as deoxyribose, C5H10O4, do not have the required ratio of hydrogen to oxygen. Deoxy means lacking a hydroxyl group, and deoxyribose is simply ribose without an OH group. Among the well-known carbohydrates are various sugars, starches and celluloses, all of which are important for the maintenance of life in both plants and animals. Thus, carbohydrates make up the bulk of organic substances on the earth. The major metabolic fuels, that is carbohydrates, fatty acids and amino acids, are degraded through converging catabolic pathways to enter the citric acid cycle and yield their electrons to respiratory chain, and how this exergonic flow of electrons to oxygen is coupled to the endergonic synthesis of ATP. The energy yielding degradation of cellular components is counterbalanced by biosynthetic processes which create and maintain the intricate orderliness of living cells. Here, biosynthesis of carbohydrates can be categorized as biosynthesis in plants, biosynthesis in bacteria and biosynthesis in animals. The first one, biosynthesis of carbohydrate in plants. The following are the important pathways. First one, photosynthetic carbohydrate synthesis. Photosynthesis in vascular plants takes place in chloroplasts. In the carbon dioxide assimilation reaction, that is the Calvin cycle, ATP and NADPH are used to reduce carbon dioxide to triose phosphates. These reactions occur in three stages. The fixation reaction itself, catalyzed by Rubisco, reduction of the resulting 3-phosphoglycerate to glyceraldehyde 3-phosphate, and regeneration of ribulose 1,5-bisphosphate from triose phosphates. Second one, Rubisco condenses carbon dioxide with ribulose 1,5-bisphosphate, forming an unstable hexose biphosphate that splits into two molecules of 3-phosphoglycerate. Rubisco is activated covalent modification catalyzed by rubisco activase and is inhibited by a natural transition state analog, whose concentration rises in the dark and falls during daylight. Third one, stromal isozymes of the glycolytic enzymes catalyze reduction of 3-phosphoglycerate to glyceraldehyde 3-phosphate. Each molecule reduced requires 1 ATP and 1 NADPH. Fourth one, stromal enzymes including transketolase and aldolase rearrange the carbon skeletons of triose phosphates, generating intermediates of 3, 4, 5, 6 and 7 carbons and eventually yielding pentose phosphates. The pentose phosphates are converted to ribulose 5-phosphate, then phosphorylated to ribulose 1,5-biphosphate to complete the Calvin cycle. Fifth one, the cause of fixing 3 carbon dioxide into 1 triose phosphate is 9 ATP and 6 NADPH, which are provided by the light-dependent reactions of photosynthesis. Sixth one, an antiporter in the inner chloroplast membrane exchanges Pi in the cytosol for 3-phosphoglycerate or dihydroxyacetone phosphate produced by carbon dioxide assimilation in the stroma. Oxidation of dihydroxyacetone phosphate in the cytosol generates ATP and NADH, thus moving ATP and reducing equivalence from the chloroplast to the cytosol. Seventh one, 4 enzymes of Calvin cycle are activated indirectly by light and are active in the dark, so that hexosynthesis does not compete with glycolysis, which is required to provide energy in the dark. Next, photorespiration and the C4 and CAM pathways. When rubisco uses oxygen rather than carbon dioxide as substrate, the 2-phosphoglycolate so formed is disposed of in an oxygen-dependent pathway. The result is increased consumption of oxygen photorespiration, or more accurately, the oxidative photosynthetic carbon cycle or C2 cycle. The 2-phosphoglycolate is converted to glycine and then to serine in a pathway that involves enzymes in the chloroplast stroma, the peroxisomes and the mitochondria. In C4 plants, the carbon assimilation pathway minimizes photorespiration. Carbon dioxide is first fixed in mesophyll cells into a 4-carbon compound, which passes into bundle cells and releases carbon dioxide in high concentration. The released carbon dioxide is fixed by rubisco, and the remaining reaction of the Calvin cycle occur as in C3 plants. In CAM plants, carbon dioxide is fixed into malate in the dark and stored in vacuoles until daylight, when the stomata are closed, that is, minimizing water loss, and malate serves as a source of carbon dioxide for rubisco. Next is the biosynthesis of starch and sucrose. Starch synthase in chloroplast and amyloplast catalyzes the addition of single glucose residues donated by ADP glucose to the reducing end of a starch molecule by a 2-step insertion mechanism. Branches in amylopectin are introduced by a second enzyme. Next, sucrose is synthesized in the cytosol in 2 steps from UDP glucose and fructose 1-phosphate. The partitioning of triose phosphates between sucrose synthesis and starch synthesis is regulated by fructose 2,6 biphosphate, an allostatic effector of the enzymes that determine the level of fructose 6-phosphate. F26BP concentration varies inversely with the rate of photosynthesis, and F26BP inhibits the synthesis of fructose 6-phosphate, the precursor to sucrose. Next is the biosynthesis of carbohydrate in bacteria. Like plants, many bacteria have thick, rigid extracellular walls that protect them from osmotic lysis. The peptidoglycan that gives bacterial envelopes their strength and rigidity is an alternating linear copolymer of N-acetylglucosamine and N-acetylmuramic acid linked by beta-4 glycosidic bonds and cross-linked by short peptides attached to the MUR2AC. During assembly of the polysaccharide backbone of this complex macromolecule, both GLC, NAC and MUR2AC are activated by attachment of a uridine nucleotide at their anomeric carbons. First, GLC NAC1 phosphate condenses with UTP to form UDP GLC NAC, which reacts with phosphoenol pyruvate to form UDP MUR2AC. 5 amino acids are then added. The MUR2AC pentapeptide moiety is transferred from the uridine nucleotide to the membrane lipid dolichol, a long-chain isoprenoid alcohol, and a GLC NAC residue is donated by UDP GLC NAC. In many bacteria, 5 glycines are added in peptide linkage to the amino group of the LIS residue of the pentapeptide is added to the non-reducing end of an existing peptidoglycan molecule. A transpeptidation reaction cross-links adjacent polysaccharide chains, contributing to a huge, strong macromolecular wall around the bacterial cell. Many of the most effective antibiotics in use today act by inhibiting reaction in the synthesis of the peptidoglycan. Many other oligosaccharides and polysaccharides are synthesized by similar routes in which sugars are activated for subsequent reactions by attachment to nucleotides. In the glycosylation of proteins, for example, the precursors of the carbohydrate moieties include sugar nucleotides and lipid-linked oligosaccharides. Next is the biosynthesis of carbohydrate in animals. The carbohydrates are an important source of carbon for synthesis of other compounds and of chemical energy. So, they function in energy storage, that is, starch and glycogen, signaling glycoproteins and glycolipids. For example, blood group determinants, fuel the nervous system and muscle, and virtually all cells. Although there are distinct cell type specific differences in choice of primary fuel molecule, are parts of nucleic acids, that is genes, mRNA, tRNA, ribosomes, and as cell surface markers and signaling in glycolipids and glycoproteins are part of connective tissue, that is heteropolymers, glucosaminoglycans. Cell wall components, that is cellulose, hemicellulose. made of polymers that are enzymatically inert for most mammals to digest, except ruminants that harbor a special digestive tract bacterium with the appropriate cellulase enzyme. The alpha-glycosidic bonds found in glycogen and starch are metabolically available to humans. Starch is the major source of dietary glucose. The enzymes responsible for starch degradation are called amylases. Other sources of glucose are sucrose, a disaccharide glucose fructose from fruits, and lactose, a glucose galactose disaccharide from milk. Only monosaccharide species like glucose, fructose, and galactose can be absorbed via active membrane transport systems. Special intestinal glucosidases split the disaccharides into their monosaccharide components. Maltose is hydrolyzed by isomaltase, that is oligo-1,6 glucosidase, and with lower efficacy by sucrase, that is sucrose alpha glucosidase. Lactose intolerance comes from a lack of lactase in many adults, causing an accumulation of milk sugar with consequences such as dehydration. In animals, especially in mammals, the major biosynthetic pathways are: first one, glycolysis, and second one, gluconeogenesis. Here is the explanation of glycolysis. Glucose metabolism is conserved throughout evolution, but species and tissue-specific variation are well known and of physiological importance. The well-studied metabolic pathways of glucose oxidation for energy uses are derived from biochemical studies of mammalian liver, muscle, and brain tissue, as well as in E. coli. Glycolysis is used by both aerobic and anaerobic organisms. Glycolysis in human and bacteria are almost identical with respect to the enzymes employed, but differ by their uptake mechanism of glucose into the cell and the end product under anaerobic conditions. In humans, glucose enters the cytoplasm through glucose facilitators, that is passive diffusion. In enteric bacteria, glucose intake is fueled by concomitant phosphorylation, while the hexose is transported across the membrane. The major pathways are glycolysis and the pentose phosphate pathway producing pyruvate. Under anaerobic conditions, the reduction of pyruvate to lactate by means of NADH oxidation is the only source of NAD4 glyceraldehyde 3-phosphate dehydrogenase. It is also known as phosphorylating. Depleting the cytoplasm of NAD effectively inhibits glycolysis. Lactate formation is the endpoint of glycolysis in mammals. The sustained use of skeletal muscle under stress is a typical anaerobic reaction, and accumulation of lactate is the cause of sore muscles after an unexpected heavy workout. Next is the gluconeogenesis. The regeneration of glucose is called gluconeogenesis and is particularly important in liver, which is the major organ involved in glucose synthesis from carbohydrate and non-carbohydrate sources. It is the only organ that can regenerate glucose from lactate. Comparing glycolysis and its reverse mode, gluconeogenesis demonstrates general rules governing metabolic pathways regarding control and reversibility. In gluconeogenesis, the glycolytic enzymes are bypassed by gluconeogenetic enzymes. Thus, gluconeogenesis involves the conversion of pyruvate to glucose, which is the reverse of glycolysis. In mammals, the balance between gluconeogenesis and glycolysis in liver has to respond to the needs of the entire body, such as mechanical work, digestion or thinking, and thus is carefully adjusted. Comparing the energy expenditure for gluconeogenesis with the gain from glycolysis clearly shows that it costs more to generate glucose from internal metabolites than to break it down once glucose is inside the cell. Here is the ending of the topic. Carbohydrates, also called carbs, are defined as aldehydic or ketonic compounds with some number of oxidrylic groups, so polyhydroxyaldehydes or ketones as well. In addition to being obtained directly from food or generated by photosynthesis, glucose can be synthesized from other organic molecules. In animal cells, glucose synthesis, that is, gluconeogenesis, usually starts with lactate produced by anaerobic glycolysis. Amino acids derived from the breakdown of proteins, or glycerol produced by the breakdown of lipids. In both animals and plant cell, simple sugars are polymerized and stored as polysaccharides. The synthesis of polysaccharides, like that of all other macromolecules, is an energy requiring reaction. Such a reaction, however, is energetically unfavorable and therefore unable to proceed in the forward direction. Consequently, the formation of a glycosidic bond must be coupled to an energy yielding reaction, which is accomplished by the use of nucleotide sugars as intermediates in polysaccharide synthesis. Glucose is first phosphorylated in an ATP-driven reaction to glucose 6-phosphate, which is then converted to glucose 1-phosphate. Glucose 1-phosphate reacts with UTP, uridine triphosphate, yielding UDP glucose plus pyrophosphate, which is hydrolyzed to phosphate with the release of additional free energy. UDP glucose is an activated intermediate that then donates its glucose residue to a growing polysaccharide chain in an energetically favorable reaction. Thus, chemical energy in the form of ATP and UTP drives the synthesis of polysaccharides from simple sugars. The biosynthesis of oligosaccharides by using the enzymatic synthesis provides a good alternative for accessing well-defined carbohydrates in sufficient quantities for the living system. Thank you.